Thank you to the organizers for allowing me to speak here today. I'm very excited to talk to you about my work. So my name is Trever Greene, and today I'm going to be talking to you all about my thesis work in the Coscoy Lab, done in collaboration with the Laurent Lab. Now, my talk is entitled Epigenetic Perturbation by a Herpes virus as a Novel Mechanism for Innate Recognition. And so, as was just alluded to, the innate system that I'm going to be talking to you about today is that of natural killer, or NK cells. Now, NK cells are innate lymphocytes that are able to directly recognize and eliminate infected or transformed cells. And they do this through a balance of activating and inhibitory receptors, which recognize unique ligands on the surface of target cells. So on a normal, healthy cell, you have a ton of ligands for the inhibitory receptors of NK cells, and very few to none of the activating ligands for these NK cells. Whereas in infection, the activating ligands can become highly induced, signaling the NK cell to kill that target cell. And so my work is focused on the regulation of these activating ligands, in particular, a family of these ligands that bind to the activating receptor NKG2D. And I'm going to be talking to you today about the RAY1 family in particular. So when we started this project, we knew a little bit about the signaling that induced these ligands, but not a ton. We knew that they tended to be induced by stress pathway signaling. And so the beginning of this project started with how do viruses induce the expression of these ligands? And so the model we used for this was MCMV, or mouse cytomegalovirus. And this is because, as you can see, MCMV infection induces the expression of the RAY1 family of NKG2D ligands really well. We very quickly, looking at this system, established that this wasn't happening through any of the known mechanisms of RAY regulation, and not through any of the known innate sensing pathways that we had, such as TLR signaling. And really, the most telling data we had at this point in the project was that this induction required MCMV gene expression. And so Maria Tokuyama, a previous graduate student in the lab, performed a forward genetic screen looking for genes that might mediate this effect. And she found that if she eliminated a single open reading frame 18, corresponding to the protein M18, that she completely eliminated the ability of MCMV to induce the expression of these ligands, and that she could rescue this by transfecting in a construct encoding M18. But the most interesting piece of data that came out of this was the fact that if she simply expressed M18 outside the context of viral infection, that she saw a huge induction of these ligands just with the protein by itself. And so this is where I came in, right when the focus of the project switched from how is MCMV inducing the expression of these ligands to how is M18 inducing the expression of these ligands. So just a quick rundown on M18. M18 is a large, uncharacterized protein. It's low complexity. From our preliminary studies, it appears to be nuclear localized, and we haven't been able to find any sequence homologs in other viruses. And so we were sort of flying in the dark here. We didn't know what we were working with. The biggest handle we had on this was the fact that it induced these ligands. And so since M18 induced the expression of these ligands at the mRNA level, we decided to see whether it could be inducing expression off of the RAY1 promoter. Now, the Laurent Lab had already narrowed down a minimal RAY1 promoter. And so what we did is we put that promoter in front of firefly luciferase and co-transfected this construct in with M18, and then compared expression of luciferase when it was co-transfected with M18 to vector control. And so as you can see here, co-transfection of M18 with the wild type RAY1 promoter induces expression off of this promoter very well. So looking for elements that might be important to this, we created deletions of this promoter. And as you can see here, deletion of 15 nucleotides had no effect, whereas deletion of 25 nucleotides almost completely eliminated this inducibility. And so comparing this to sequences of transcription factor binding, we were able to show that this was very homologous to the binding sequence for SP1 and SP3 transcription factors. So taking a look in vivo at chromatin immunoprecipitation, we saw that SP3 was actually the particular transcription factor occupying this site. But M18 didn't appear to change this occupancy at all. And so this helped us start to build up our model. M18 was definitely acting through this SP3 binding site, but it didn't appear to be interacting with SP3 binding to this site. So looking into the literature, we found that SP factors often regulate genes through the recruitment of the transcriptionally repressive histone deacetylase enzymes, or HDACs. And this is really important because it had been previously established in the Raleigh lab as well as other labs that chemical inhibitors of HDACs are strong inducers of NKG2 ligand expression. And you can see that here with the inhibitor butyrate, you get a strong expression of RAY1 as compared to vehicle control. And so we wanted to see if perhaps this SP binding site was important for this. And we did the same luciferase assays I described before, except with butyrate as a stimulus instead of M18. As you can see, the wild-type promoter is induced incredibly strongly by butyrate treatment, whereas a mutant of this SP factor binding site is essentially completely abrogated in its ability to be stimulated by these chemicals. And so this helped us add another piece to our model here. HDACs, this SP site appears to be important for stimulation of this promoter by HDAC inhibition. And so since it was important for HDAC inhibition, it was important for M18, we thought perhaps M18 is actually acting through HDAC inhibition. And a very simple prediction of that very simple model is that acetylation would increase in cells expressing M18. And my Western blot is completely blank. So you can come visit my poster or take my word for it, but something has shifted around here and you can't see the bands on the acetylated H3, which should be incredibly increased. So please visit my poster for a much more convincing version of this blot. So in the blot you can't see is very suggestive that M18 is interacting with HDAC regulation. So this still left the question of how. And so our collaborators in the Deacetylace lab helped us do an IP mass spec experiment in which we identified interactors of M18. And we found one particular interactor that might help explain the connection between M18 and HDACs. And that's the kinase CK2. And OK, something is really wrong with how this transitioned, because this Western blot is wrong too. So the kinase CK2 is an activator of histone deacetylases. And as you also can't see, M18 interacts with it. And so in order for M18 to be acting through CK2 on HDAC regulation, you'd expect that M18 would be inhibiting this activator of HDACs. And so as, what I'm showing you here is levels of phosphorylation of HDAC1 and HDAC3 at the CK2 phosphoconsensus sites in M18 expressing cells. And so as you can see, these are severely decreased, which is exactly what we'd expect in this model. And so the whole model put together, in a normal, healthy cell, CK2 is phosphorylating HDACs, enhancing their activity and repressive potential, and RAY1 expression is repressed. Now in MCMV infection, M18 inhibits CK2, which leads to less phosphorylation and less activity of these HDACs, de-repression of this promoter, and active transcription. And so what I find really interesting about this system is that we have an innate immune molecule that has an HDAC-sensitive switch in it. that is able to recognize HDAC inhibition as a metric of infection. And HDAC inhibition isn't limited to MCMV. Lots of DNA viruses do this. And so to expand these observations, we took other HDAC inhibiting viral proteins and we put them into human cells. And we looked at the expression of human NKG2D ligands. So what you're seeing here is transfection of HFFs with the HDAC inhibiting ICP0, IE1, and IE2, as well as a control EBNA1, which does not inhibit HDACs, but is a nuclear localized low complexity viral protein. And as you can see, the expression of the human NKG2D ligand, ULBP1, is significantly in ICP0 and preliminarily in IE1 and IE2 induced by expression of these HDAC inhibiting proteins. And so what I find really interesting about this is that it suggests that epigenetic perturbation by viruses is such a fundamental piece of their life cycle that the immune system has developed a way to sense and respond to it. And so I'll take questions in just a second. You can come see my poster afterwards for all of that data that wasn't in this presentation. But first, I would like to acknowledge everybody who's helped me with this. Maria Tokoyama, Michelle Kuntz, and James Lin, who actually did experiments I showed you earlier in this talk. Our collaborators at the Deacetylace and Deacetylace Lab, David and Laurent for being excellent mentors, and then everybody in the Glonsinger and Coscoy Labs for all of their support through this project. And then finally, everyone who gave me reagents to help make this project happen. Thank you all. And now I'll take questions.
Questions, comments?
Very interesting. I was wondering on the chip assay, when you had M18 in there, you had less SP3 accumulating on your promoter. Is that just this particular experiment?
That's that particular experiment. I've done that 4 or 5 times, and it's sort of, they don't really even out to being less.
It could still be a combined effect that you have there, but
yeah.
It definitely looked reduced when you looked at the y-axis.
Yeah, yeah.
Thank you.